Hello everyone, welcome to MR Learning Circle. I hope you all are doing good. From today, I am going to start a series of video lectures for microbiology. This series will be beneficial for your GATE exams, CSIR NET exam, and for your academic. For update regarding the videos, please subscribe to the channel and click the bell icon. And you can also join the Telegram channel and follow on Instagram for any queries. Now let's start with the first lecture, which is about the discovery of the microbial world. So, what happened? Earth was formed 4.5 billion years ago. And what happened within the next billion year? The first cellular life form, which is known as microbe, appeared. That means microbes were the first organisms which appeared after the formation of Earth. Then, since that time, microorganisms have evolved and diversified. These microorganisms have evolved and diversified, and they occupy virtually every habitat of the earth, like from the oceanic geothermal vents to the coldest Arctic ice. You can see the diversity. It is even living in the geothermal vents, and it is these microbes are even present in the Arctic ice. Recently, the scientists in Russia have revived a microbe. You can say they named it as zombie virus. The link of the video is given, will be mentioned somewhere above. You can just find the video and know more about this zombie virus. So, after that, this diversity of the cellular microorganism is best exemplified by their metabolic capabilities. So why are these microorganisms diverse? Because of their metabolic capability. Metabolic capabilities of the microbes make them diverse. So a microorganism name Prochlorococcus, it is a cyanobacterium. Formerly it was known as blue-green alga. It was the most abundant photosynthetic organism. Question could be asked, okay, in your examination, that which was the microorganism or cyanobacterium which is the most abundant photosynthetic organism. So if you would know the name, it would be easy for you to answer. And you should also know that it is a cyanobacterium which was formerly known as blue-green alga. Clear? Now the members of the microbial world. So what are the organisms the microbiologists have to study? So the organisms of biological entities which is studied by microorganisms are classified as cellular organisms and acellular organisms. So the cellular organisms include the fungi, the protists, the bacteria, and the archaea. And the acellular organisms include virus, viroids, satellites, prions. So this section is slightly more important. You could, in this you will get question like viruses are composed of proteins and nucleic acids. So you could get the answer that question that what are viruses are composed of. Options would be given A, B, C, D. Then if you know that it is composed of protein and nucleic acid. Then viroid is composed of RNA. Similarly, satellite is also composed of nucleic acid only, and that too often RNA. That means mainly RNA is present or in the satellite, and prions, protein. Clear? That was about the classification. Now the microbial world. So in this microbial world, you will know about the definition of microorganism. Yeah. So microorganisms are those organisms which are acellular biological entities. That means they are not cellular, they are not made up of a particular type of a cell, and they are too small to be clearly visible by an unaided eye. That means you cannot see it with your normal eye and they are not visible. But although the small size is an important characteristic of the microbe, it's alone not sufficient to define them, because some cellular microbes such as bread molds and filamentous filamentous photosynthetic microbes, they are visible without microscope. So they are termed as macroscopic microbes. So these macroscopic microbes are often colonial. Colonial means they live in a colony or they form a colony, and consisting of small aggregation of cells. So small aggregations means a group of cells, small cells, and then they form a colony. Then this is a microbe and then they will form a colony. Some macroscopic organisms are multicellular. Now, how are these multicellular microorganisms distinguished from plants and animals? So, the microorganisms who are multicellular lack highly differentiated tissues. They lack highly differentiated tissues. That means the animal and the plant group have differentiated tissues, whereas the microbes who are multicellular do not have highly differentiated tissues. Very important difference. Just note it in your notebook. Now, this diversity of microorganisms have always presented a challenge to the microbial taxonomy. So now let us learn about what is the prokaryotic cell and what is a eukaryotic. First of all, prokaryotic. Prokaryote is a Greek word which means pro means before and karyon means nut or kernel. So the organism with a primordial nucleus. The main important point is that they lack a membrane delimited nucleus. Membrane delimited nucleus means the nucleus is not bound by a particular or a well-defined membrane. Okay, and their contents, the content present in the cell, are not divided into compartments by the membrane. These cells are not bound by any of the membrane in the compartment. This thing is not present. Okay, now about the eukaryotic cells. So the eukaryotic cell, also a Greek word, eu means true and karyon means nut or kernel. So they are membrane bound nucleus. They have membrane bound nucleus as well as the cell organelles which is present in the cell. And they are separated from cellular material and processed from others. Now, these observations eventually led to the development of classification scheme that divided the organisms into 5 kingdoms. That is Monera, Protista, Fungi, Animalia and Plantae. So, microorganisms, except for viruses and other acellular infectious agents, which have their own classification system. These viruses and acellular organisms have their own classification system. So all the microorganisms except them were placed in the first 3 kingdoms, that is Monera, Protista and Fungi. This 5 kingdom system was an important development in the microbial taxonomy, but it is no longer accepted by the microbiologists now. Because why this is not accepted? Because not all the prokaryotes are same. All prokaryotes are not same. Okay, and they should not be grouped. So they should not be grouped together. in a single kingdom. So the three areas that profoundly affect the microbial classification is that first is the they very much thing have been learned about the detailed structure of microbial cells. That means the detailed structure of microbial cells from the use of electron microscopy. With the help of electron microscopy, now the microbiologists are able to get the detailed structure of the microbial cells, which have changed many things. Second, microbiologists have determined the biochemical and the physiological characteristics of many different microorganisms. By performing different biochemical and physiological tests and experiments, they are able to characterize them differently. And third and the last is reason is sequence of nucleic acids and protein from variety of organisms have been. That means the nucleic acids of microorganisms, plants, animals, and many other organisms have been compared. So this has made the microbial classification slightly complicated and confusing. So now what happened next? Later based on rRNA comparison. Studies based on rRNA comparison started. So the comparison of ribosomal RNA, that is rRNA. So now what are these three modern cells which had three different roles of catalyst, structure molecule and hereditary molecule? These modern cells are proteins, DNA and RNA. So proteins have two major roles. Proteins have two major roles. One is the structural role and the other is the catalytic role. So this catalytic protein are called enzymes and they either speed up the myriad of the chemical reaction that occur in the cells. Now what does DNA do? DNA stores hereditary information and can be replicated to pass the information to the next generation. From generation A to generation B, this DNA replicates and passes the hereditary information. Now let's come to the RNA. RNA helps in converting the information that is stored in DNA into protein. Here it is used to convert the information that was stored in DNA into a protein. Now coming to the RNA world hypothesis. So how did this RNA world hypothesis? Any hypothesis about the origin of life must account for the evolution of these molecules, where protein can do the cellular work, but their synthesis involves other proteins and RNA and uses information stored in DNA. And DNA can't do cellular work. It stores genetic information and serves as a template for its own replication. RNA synthesis using DNA as a template and protein as a catalyst for the reaction. So these three modern cells were interdependent on each other. But we learned earlier that it was a single cell which would be able to perform the three functions of storing, converting, and replicating everything in a single molecule. All the quality should be present in the single molecule. Now, what happened was that So based on these consideration, it was hypothesized that at some time in the evolution of life, there must have been a single molecule that could do both cellular work and replicate itself. That was the thing I explained to you earlier. Now, what happened in 1981? Thomas Sesh discovered a catalytic RNA molecule from a protist named Tetrahymena species. And this catalytic RNA could cut out an internal section of itself, internal section of itself and splice the remaining section back together. What did it did? It could cut. That means, for example, just take this molecule and this would cut this catalytic RNA could cut it and splice out a part of it, and after that it could combine or it would join the remaining part. So what was that? Since then, other catalytic RNA molecules have been discovered, including an RNA found in ribosome that is responsible for forming peptide bonds. Now, what are these peptide bonds? These peptide bonds are used to bind together the amino acid to form a protein. So, this catalytic molecule are now called as ribozymes. So this catalytic RNA is now known as ribozyme. Clear? Now, after the discovery of ribozyme suggested that RNA at some point had the ability to catalyze its own replication using itself as a template. RNA used itself as a template. Now, in 1986, what happened? In 1986, Walter Gilbert coined the term RNA world to, to describe the pre-cellular stage in the evolution of life. This RNA world used to describe the pre-cellular stage in the evolution of life, in which RNA was capable of storing, copying, expressing genetic information, catalyzing other chemical reactions. However, for this pre-cellular stage to proceed to the evolution of cellular life forms, a lipid membrane must have formed around the RNA. A lipid membrane should be present around the RNA to proceed the evolution of this pre-cellular stage of evolution of life. Now, a fascinating experiment performed by Marin Hanks, Shelley Fujikawa, and Zach Zostek in 2003 showed that clay triggers the formation of life. That it is the clay that triggers the formation of liposomes that actually grow and divide. Now, together with the data on ribozymes, these data suggest that early cells have been liposomes containing RNA molecules. So, what was the conclusion after many experiments? That liposomes may have been the early cells which contained RNA. Clear? Now, apart from its ability to perform catalytic activities, RNA perform the function of RNA suggests its ancient origin. Apart from it, the RNA has some ancient origin. Consider that much of the cellular pool of RNA in modern cell exists in the ribosome. Now consider it the most of the cellular pool of RNA exists in ribosome. And this ribosome is a structure that largely consists of rRNA, and it uses mRNA to construct the mRNA and transfer RNA to construct the protein. Now, thus RNA seems to be well poised for its importance in the development of proteins. Because RNA and DNA are structurally similar, RNA could have given rise to the double-stranded structure of the DNA. It is also suggested that once DNA evolved, it became a storage facility for the genetic information. Why? Because of its chemically stable structure. DNA is double-stranded and it is more stable than the RNA. You must know. You all must be knowing this. So this stable structure was the main reason it became the storage facility for the genetic information. Now, two more pieces of evidence that support the RNA world hypothesis is that the fact the energy currency of the cell, that is ATP, is a ribonucleotide. Okay. And the other evidence is that RNA can regulate gene expression. RNA could regulate gene expression. So two more evidence is that ATP is a ribonucleotide and RNA regulates gene expression. So it would seem that the protein, DNA, and the cellular energy can be traced back to the RNA. Clear? Thanks for watching. That was all for today. I hope this video gives you something important and you get benefit in your examination as well as in your life. Thanks for watching. Take care. Bye.